Hello, I'm Professor Peter Nash from the Griffith University in Brisbane, and today I'm joined by Professor Ebina from the Osaka University in Japan. And we're very grateful, Professor Ebina, for your time. We know how busy and difficult the time is at the moment. Today we'll be studying a study recently published in Arthritis Research and Therapy, which analyses drug retention using the ANSWER registry, which is based in the Kansai district of Japan. This study compares 7 biologic disease-modifying drugs as well as the targeted synthetic tofacitinib in both biologics-naive and biologics-switched patients with rheumatoid arthritis. Welcome, Professor Ebina. Can you tell us a little bit about yourself, your research interests, and your hospital, please?
Okay. First of all, thank you for providing me a great opportunity to introducing our study. So my name is Kosuke Ebina from Osaka University, and my interest, I'm a rheumatoid orthopedics doctor, and in Japan, orthopedic doctor also prescribes biologics or JAK inhibitors for rheumatoid arthritis patients. My specialty is foot and ankle surgery, and also my research interest is clinical rheumatology and osteoporosis and the regenerative medicine of the osteoarthritis or the meniscus.
Well, that's very impressive because no orthopedic surgeon in Australia would ever be interested in any medications or biologics or prescribing. They just want to operate.
Yes,
so
I understand.
It's a pleasure to have you. So
Yes.
Can you tell us a little bit about the Kansai district of Japan, please?
Yes.
And the ANSWER registry in particular.
Yes. Our ANSWER cohort is a multi-center observational registries, including 7 universities affiliated hospitals in the Kansai district of Japan, which was developed in 2015. The unique point of our cohort is including both rheumatologists and orthopedic doctor, because orthopedic doctors also prescribe biologics in Japan. We are retrospectively and prospectively updating about 10,000 RM patients' data every year.
And roughly the population of the Kansai district, how many people do you drain?
The living populations, you mean?
Yeah.
So I'm sorry, I'm not sure, but the Kansai district is second largest district. From the Tokyo is the capital of the Japan, and the Osaka is the second biggest city in Japan. So the
many millions.
Very many millions, very
That's
big
right.
populations. Yes.
Okay, and I know that in our country, it's very difficult to get our rheumatologists to enter any data in any registry because
really?
they think they're very busy.
Yes.
So how do you get your rheumatologist to enter all this data? Is it done, are they paid to enter? Is it done out of goodwill? Is it done by the office staff? How is it done?
Thank you for your question. It's mainly the doctors is taking part by their own will to provide the new evidence for improving the rheumatic diseases. And now we have some sponsor from the companies to help the data from the other specialists, not for the medical doctors. So we are using these companies' help for updating the data. Yes.
Excellent. And can you tell us a little bit before we start talking about your trial, how the COVID-19 has affected practice in Japan? Do you do a lot of telemedicine? What has happened, the situation in Japan?
Okay. The number of patients with COVID-19 is still increasing in Japan. And some patients prefer telehealth. about 10 or 20% of my patients. And some patients are worried about continuing biologics or JAK inhibitors, although I recommend them to continue present treatment if they are well controlled in accordance with the Japanese colleague of rheumatologist's recommendation.
Excellent. So just tell us a little bit about this particular study and how you went about doing this study.
I previously take part in the drug retention in Osaka University in our institution, but I wanted to investigate for many patients' data, and our ANSA cohort has about 10,000 RM patients' data and very precise data. So I think that I can provide more useful information for the drug retention of biologics using this cohort.
Okay. And with the biologics in Japan, can you give us a feel for how they are used? Are they used very commonly or very rarely? Because Japan has rules that they use a very low dose of methotrexate. What, compared to the rest of the world, what do you think about this methotrexate restriction?
Okay, indeed, many Japanese have restriction in MTX dose due to drug hepatopathy or nausea. However, recent study demonstrated that intra-erythrocyte MTX polyglutamate concentrations, which are considered as a useful biomarker of MTX efficacy, were more than 1.5 times higher in Japanese compared to Western populations. So I suppose that relatively low dose of MTX is also effective for Japanese. And in Japan, about 30 or 40% of RA patients is treated by biologics or JAK inhibitors now, because we have a public health insurance by the government, and we have to pay about 10 to 30%, only 10 to 30% of the payment by ourselves. So maybe this promotes, yes.
And are they all very much similar priced? There's no advantage in a JAK over an injection, or they roughly the same like they are in our country?
Oh, okay. In Japan, for example, the cost of tocilizumab is about half of other biologics. It's very cheap. So this may influence the prescription number in this cohort.
And how have the JAKs gone as a class in Japan?
So thank you for the question. The JAK is, we have 4 JAKs in Japan, and we can prescribe.
They're doing quite well?
Yes, but the prescription number is not still so high, maybe about 5 or below 10%, yes.
So quite small.
Yes.
In our country, we're seeing more and more biosimilars, but the uptake is still very low. What's it like in Japan?
Yes, we have 2 biosimilars in Japan now, infliximab and etanercept. And recently, prescription number of etanercept biosimilar is rapidly increasing. It's very increasing in etanercept.
Okay, so tell us how you performed this study and a little bit about the patients selected.
So, in general, all of the RA patients fulfilled 1987 ACR or 2010 ACR EULA criteria were included in this cohort. And in this paper study, patients who were treated with biologics or tofacitinib from 2001 to 2019 with data on starting and discontinuation dates and reasons for discontinuation were all included.
Okay, and how was drug retention compared in this study?
As I mentioned, drug discontinuation reasons were categorized into 4 major reasons: including lack of effectiveness, toxic adverse events, non-toxic reasons, or remissions, and estimated at 36 months using the GRAY test.
Tell us a little bit about the GRAY test, not familiar to many rheumatologists.
Sorry. It is used for the calculate the cumulative discontinuation ratio for the other. It's popular in other drug studies, and if we wanted to adjust their clinical backgrounds, we can use the fine gray model to adjust it by the potent confounders, as I mentioned. We use the both models.
And what were the main findings from the study?
Yes, the main findings of this study was there was a remarkable difference in the drug retention rates of the 7 biologics and tofacitinib between the biologics-naive and switched cases. And the overall retention rates were highest in abatacept among the naive cases, and the tocilizumab showed the highest retention in the switched cases.
Okay, and any reasons for that, do you think?
Yes, I think there are some limitations to be considered. First, as I mentioned, the combined MDX usage may strongly affect the TNF inhibitors compared to the non-TNF inhibitors. So the prescription ratio and the dose was relatively small in this cohort, so it may make advantage for the non-TNF inhibitors. And the other reasons is there's a high-aged rheumatoid arthritis patients in Japan. It's still increasing, so there's a trend to be treated by the abatacept because they have many lung diseases or the renal dysfunction.
Yeah.
Yes, so the physicians tend to use abatacept or the etanercept, which has the evidence of the low toxic adverse events. Yes.
Okay. I was thinking about the TNFs. Is TB still an issue in Japan? Is it common?
Thank you for your question. The patients who have the active TB is not so familiar in Japan now.
Sure.
But
sure.
Some patients have positive quantiferon test, and we have to treat them.
Be careful.
Yes, be careful. Yes, but not
so.
So
What were not so common anymore. What was the most common reason for discontinuation?
The most common discontinuation reason was the lack of effectiveness. It's the primary effectiveness or secondary, yes.
Okay. And it seemed unusual that Cimzia, certolizumab, seemed to have the worst discontinuation and the most adverse events. Is that really a real finding, do you think?
Yes, there's a limitation because the number of patients treated with certolizumab was relatively small. But for my impression in Japan, there are a little bit high rate of patients of lack of efficacy in certolizumab pegol. So from our data, we showed that the patients who were treated with certolizumab pegol for more than after the third biologics, they have very low retention. So certolizumab pegol should be used in first or second biologics, maybe.
Not third. And can you comment on how tofacitinib compared to the other biologics?
This was the most interesting point. So tofacitinib showed higher retention due to lack of effectiveness next to tocilizumab, although showed lowest retention due to adverse events. So in short, it is effective, but it maybe have a low retention due to adverse events, such as herpes zoster. Yes.
I was going to say, what how did the zoster issue get handled in Japan?
Uh,
Because your rates of zoster are higher than other countries in the world.
mhm. We have, of course, medication for the herpes zoster. So we educate patients that JAK inhibitors have a relatively high rates of herpes zoster. So if you have a symptom, go to the hospital as soon as possible to be
Right.
prescribed with the treatments,
And do
drugs.
you have vaccination for herpes
Uh,
zoster?
yes, we have vaccination, but not so much popular still.
Right.
So because it is very expensive still now and
Yeah.
very painful, yes, for
And
patients.
do you have the killed vaccine Shingrix, or do you only have the
Yes,
live?
we have Singwix, yes, now.
Okay, okay. Because we can't get it in Australia.
Oh,
They
really?
don't. They don't make enough to supply America and Canada, leave alone Australia.
Okay, I understand,
Okay.
yes.
From one of the tables, the remission
Mhm.
rate was really quite low,
Yes.
1 to 3 to 10%. Do you
Mhm.
think that's accurate? Do you think more patients are in remission that are recorded in the
Mm,
registry?
yes. Uh, from our data, the patients who discontinued by remission was highest in infliximab and
Yeah,
adalimumab for the
yeah.
biologics-naive patients because these two agents have many evidence that they can stop the treatment by previous studies. And in general, Japanese physicians do not, they're not so uh, they are not so interested in discontinuing these biologics, but they prefer to the dose, the reducing the dose or the expanding the treatment. Uh, how should I say? The expanding the treatment.
Yes. What would you say was the common causes of discontinuation?
The most common cause was the ineffectiveness of the
Mhm.
treatment. It is similar to the other previous studies. About 50% of the discontinuation was due to ineffectiveness.
And do they use an objective measure like CDI or dash 28?
Mm,
What is used in Japan?
yes, we of course use DAS or CDI, but in this study, it depends on the each physician's decision. So there was
Right.
no criteria for the discontinuation. Yes.
Okay. And what do you think was the factors that led to very good retention
Mhm.
with different biologics? Could you tell from the study?
Mhm. So there are many limitations for this study because this is an observational study. But for my opinion, uh, tocilizumab is very effective in Japanese population with or without methotrexate. And we have
Yes.
different formula like subcutaneous or intravenous. So maybe this data of tocilizumab reflects the Japanese data. And for the abatacept, because if patients has many comorbidities such as lung diseases, so we there's some populations we cannot select uh, the biologics instead of abatacept. So this may be the very important bias for selecting the treatment. Yes.
Yes. It has a perception of being a safer biologic,
Yes.
particularly in the setting of infection.
Mm-hmm.
Um, can you tell us then the take-home message for the
Mm-hmm.
practicing clinician? Is there
Mm-hmm.
any biologic you'd recommend based on this study, or you don't think it's appropriate to recommend?
Uh, uh, I cannot strongly recommend uh, the specific agent from our data, but if are you can provide the patients for the several agents for the first choice of the treatment or the second choice of the switched patients, because there are some similar retention agents, as you can see in our paper. So not for the one agent, but you can see the several treatment choice for the patients. Then
And
if you have
which would you, sorry, keep going.
Okay, it's okay. It's okay.
Which would you say is the safest biologic or JAK?
Yes, for the treatment naive patients, maybe abatacept has safety, and for the switched patients, etanercept have a safety profile, maybe. Yes.
That's very interesting because every registry shows that etanercept has the best drug survival amongst the TNFs. And that's a surrogate for still doing well and not having adverse events. So that fits with what we see in other registries. What about corticosteroids? Is that used very widely in Japan? And did it have a negative effect?
Yes. In Japan, the prescription ratio of the glucocorticoid is decreasing, but still about maybe 20 or 30% are prescribed with glucocorticoids, and they have a negative effect on the retention of the all biologics. So if you want to have a good retention, the glucocorticoid may be reduced in using the biologics. Yes.
So less adverse events. So thank you very much again for your time, Professor Ebina. This has been the CSF Author Interview Podcast. If you would like to know more about this paper and others uploaded to the CSF website this month, you can get detailed slide sets available in the publication section at cytokinesignaling.com. Please subscribe to this podcast on iTunes or wherever you get your podcasts from, and let us know what you think. We would love some feedback. So thank you very much, Professor Ebina. We appreciate your time. Thank you.
Thank you.